So, as we discussed in our last lecture, that the complex diseases are caused because of the multiple factors. It could be genetic, it could be environmental, it could be lifestyle factors. So, what happened in case of the complex diseases, if we talk about, usually what happened that in the case of complex diseases, single gene is having very small effect. It's having a smaller effect. And multiple genes altogether they are creating a problem. For example, if we talk about liver cancer, so it's not just one gene that is causing this disease. There are multiple genes, and each gene is having only smaller effect. That is the complex diseases. What happened in case of the linkage analysis? You find out, you know that a particular gene is creating a problem. For example, in case of the cystic fibrosis, what happened in case of the cystic fibrosis, you know that this is the gene, this is creating a problem. So this gene is having a very larger effect. So you can find out the linker molecule, and you can say that this particular gene is responsible for causing a disease, or this particular gene is linked with the particular gene that is causing a disease. So, but in case of the complex diseases, you need to you cannot perform the linkage analysis very nicely, and the reason is this, that the linkage analysis always depend on the LOD score, and whenever the LOD score, as I discussed in my last lecture, it is more than 4, you can say that there is a linked there is a linker molecule that is linked with the always linked with the particular gene that is causing a disease. But if it is less than 4, you can say the linkage is not sufficient. Linkage is poor. Otherwise, if it is more than 4, you will say linkage is strong. But what happened in case of the complex diseases? Because each gene is having very smaller effect. When you calculate the LOD score, just let me highlight it. When you calculate the LOD score in case of the complex diseases where single gene is making a very smaller effect and multiple genes are making a larger effect, in case of the complex disease, then you will never find the LOD score more than 4, and you will say, okay, this gene is not having any effect. So that's how you miss the disease causing gene. Okay. So one more time, I want to clarify you. There is a linker molecule. There is a disease causing gene. You find the linkers that is associated with the disease causing gene, and you know if this particular this, and you find out the trait. Always you will find that this linker molecule is going to the next generation with this particular disease causing gene. So you can identify the trait, how it is moving in the families, if you know this linker molecule. But in case of the complex diseases, single gene is making only smaller effect. So in that case, if you calculate the LOD score, you will never find the LOD score that is more than 4. Only very in a very rare cases you will find, otherwise you will find the LOD score less than 4. So even though you find the linker, you will say no, this linker is not associated with this. This gene is not going with this link. This gene is not linked with this linker molecule, and that's how you miss the disease causing gene, even though it is contributing to the disease. And that is the major problem of the complex diseases, because linkage analysis is always a statistical method. You have to calculate the LOD score, and once you calculate the LOD score for the complex diseases, in case of the linkage analysis, in majority of the times you will find the LOD score less than 4. In that case, you cannot say that this particular linker molecule is associated with a linked disease causing gene. It means this gene is not causing any disease, and that's how you missed that gene. So I hope you understand. So majority of the susceptibility genes turned out to have a small effect, means single gene is having only a small effect. Diseases are basically caused because of the multiple genes. It's a polygenic in nature, complex diseases. So linkage studies lacked the power, means it's a statistical method, and because of this you cannot calculate the LODs. But you, even though you are calculating the LOD score, that is not accurate, and you cannot rely on that. So what happened in 1996? New technology came. That was the association study. So what it is doing basically, it is doing, suppose this particular allele is 36% of the general population, but in case of the rheumatoid arthritis, it is 78%. Then you will say, if you are finding HLA-DR4 allele in a particular individual, you can say you that you might be susceptible for the rheumatoid arthritis. So this is a strong association suggesting that HLA-DR4 is involved in RA susceptibility. It's simple as that. So association studies can detect small differences in the allele frequencies between affected and the unaffected pupils, even when the genes affected modest. So this is the association study. So this is a definition of linkage, how linkage works. It's a genetic association between two locus positions on chromosomes. Locus that are physically close on the same chromosome tend to be inherited together during meiosis. If a disease, example is, if a disease gene is close to HTR4 on the X chromosome, family members with the disease may also share the HTR45 allele along with the disease causing gene. Coming to the association again, this is a statistical relationship between an allele and the phenotype. If the allele appears more often in people with the disease, for example, HLA-DR4 is more common in people with the rheumatoid arthritis, and if you are getting in particular individual this the presence of this allele, you can see you might be susceptible for the rheumatoid arthritis. So this is the association studies. So what is the difference? Linkage is about inheritance pattern, how the DNA segments travel through families. Association is a statistical correlation, how alleles and traits show up together in the population, how they are associated, whether the particular gene is associated with a disease or not. However, linkage, how will you find it out? How it is traveling in the families. Okay. Like in this case, you can say this particular gene, disease gene is close to this allele. So this allele you will find in the family members who are suffering with that disease. So that is how DNA segments travel through families. But you know, GWAS, genome-wide association studies, they have certain limitations because they overlook the CNVs. CNVs are, if you are not aware about the CNVs, please go ahead and look my previous lectures. I have explained them in the DNA polymorphism lecture. So CNVs, basically what happens, these are the copy number variants. So either the whole gene we are talking about, or we are talking about a larger segment of the gene. And CNVs, if it is 0, it is absent. If it is 1, there is deletion. If it is 2, you can say normal. If it is 3 or 4 or 5 or 6, you can say that is multi-allelic. And when we are saying about the biallelic nature, it means either the CNVs are present or they are absent. So what happened in case of GWAS? GWAS basically calculate the count the SNPs. And sometimes the SNPs, they do not tag the CNVs, specifically when they are in multiple copies, like 3 copies or 4 copies or 5 copies or 6 copies. Then in that case, SNPs are not tagging them. SNPs are not uh, associated with them. So what happened? They ignore the CNPs. So what you will get in your data, okay, I did the SNP and I did not find any result. I mean, the negative result. There is no SNP in that person that is related with the disease. However, that person may have the CNVs. So that is overlooked in the genome-wide association studies. They overlook the CNVs. They do not count the CNVs because sometimes the SNPs are not tagging the CNV. So let's say CNVs are overlooked. They are fewer in number, but they cover more base pairs than SNPs. As I said, either the whole gene or a larger segment. Some CNVs affect entire genes, leading to real observable phenotypic trait. For example, the number of salivary amylase gene copies affect the digestion. How well do SNPs capture CNVs? In some families, they capture CNVs pretty well. For example, in non-African families, about 50% of the biallelic CNVs, either they are present or they are absent, were tagged well by SNPs. While in case of the African population, this is just 22%. In case of the multi-allelic CNVs, where there are more than two forms, means there are multiple copies, 3 copies, 4 copies, 5 copies, they are poorly tagged by the SNPs in all population. Does this matter? If the disease is caused by the CNP, then the CNP is not well tagged by the SNP, and DWAS might miss the association entirely. What you will say, okay, I did the genome-wide association studies, I did not find any variant in this particular individual that can cause the disease. But there might be CNVs that is ignored. So early CGWAs may have overlooked important disease-causing variations. But with newer technology, this gap could be filled. For example, if you are doing whole genome sequencing, if you are doing CNV specific calling algorithm, long read sequencing, structural variant aware arrays, these directly measure the CNV. So you have to use the technology which is directly measure the CNVs instead of guessing by a CNP, SNPs. So now this is like mutation. How many kinds of mutations are there? For example, nonsense mutation when they are introducing stop codons, frameshift mutations, splice site mutation, they disrupt the normal RNA splicing, deletions, missense mutations. And whenever these kind of mutations are there, they code for the non-functional or unstable proteins, and they may trigger the nonsense mediated mRNA decay. So what is nonsense mediated mRNA decay? We talk about, suppose this is your mRNA, and suppose there is a stop codon comes all of a sudden, and mRNA length is this one because of the stop codon. This is the stop codon. Then this particular mRNA is recognized by certain proteins, and these proteins will degrade this premature mRNA. So this is the a nonsense mediated mRNA decay. Why it is called nonsense? Because there is a nonsense codon, stop codon, that comes early in the mRNA transcript, and because of if this mRNA is translated to the protein, your protein is going to be truncated protein, non-functional protein. So what these proteins will do? These proteins will recognize this mRNA and will degrade it. So there are certain examples. For example, cystic fibrosis, CFTR is mutated, DMD, DMD gene is mutated, like this. So I think I missed one slide here. Yeah, this slide. So pathogenic DNA variants are changes in the DNA sequences that leads to the diseases, and that will produce the protein which is not functional. They are impacted broadly classified into two main categories. One is the loss of function mutation and one is the gain of function mutation. So gain of function mutation means the mutation that is giving some kind of gain. For example, that will increase, change, or confers a new function to the protein. Common types is missense mutations. These mutations create hyperactive or novel protein. Gene duplications, the gene is duplicated. So again, there is a gaining lead to protein overexpression. Regulatory region mutations, they might upregulate the expression of a particular gene. What is the mechanism? The protein acquires a new biochemical activity or becomes constitutively active and often escapes normal cellular control. For example, if you see the in case of the Huntington disease, CAG repeat expansion. If number of CAG repeats are 50 in normal individuals, in case of the Huntington disease, it is more than 500, something like that. So it's in case of cancers, gain of function mutation in KRAS gene, BRAF gene, EGFR gene. So what is the phenotypic consequences? Typically dominant inheritance in case of the gain of function mutation can cause the toxic effects, ectopic activity, or cellular overgrowth. So by the term only you can understand gain of functions means that protein is gaining the excess of function, and because of that it becomes toxic. For example, CAG repeats are 50, it should be 50, but if it is 500, that is giving this particular disease. If there is a mutation in these kind of genes and these genes are overly expressed, then they are leading to cancer. So that is again the toxic behavior. So if you differentiate between the loss of function and gain of function, protein activity is lost in loss of function, is increased, or new activity is gained in gain of function. Inheritance is often recessive in case of loss of function. In case of gain of function, it is often dominant. Common mutation type is nonsense, frameshift, deletions in case of loss of function. In case of gain of function, it could be missense, duplications, promoter mutations. Examples are CFTR, PAH in phenylketonuria, FGFR3, HTT in case of the Huntington disease. Effect on health, loss of essential functions. In case of gain of function, it is abnormal or toxic waste. So this is the difference between the loss of function and the gain of function mutations. Understanding whether a mutation leads to loss or gain of function is crucial for predicting the inheritance pattern. For example, in case of LOF, you can say it is recessive, or in case of gain of function, it might be dominant. Design the targeted therapies for the gene replacement for the LOF, inhibitors for the gain of functions. If suppose there is a loss of protein function, you can replace the entire protein by the gene replacement method. Genetic counseling and diagnostics, it is also very necessary. So these are about the loss of function and gain of function mutations. What is the difference and why it is important? So that's all about today's lecture. Thank you.